And then, but first, I'll talk about why we're interested in light microscopy and so in fluorescent microscopy, two commonly used volumetric imaging methods are confocal and light sheet microscope. So both the systems are scanning based and directly obtain a 3D information object, either by scanning the volume point by point or plane by plane. While these strategies renders high resolution images, and they are inherently slower and most are not suitable for imaging moving structures such as the heartbeat or fast neural dynamics. So light field microscope, on the other hand, acquires the 3D information in a single shot without scanning, and use computational methods in the post-processing to recover the 3D volume. Although compared to confocal and light sheets, light field has less resolving power, meaning less resolution, than in extreme. The volumetric imaging speed will increase significantly, which is very powerful for capturing fast dynamics changing in 3 dimensions. So how does light field achieve that? We know that for the 2D conventional imaging system, such as a wide field microscope, all the light rays from a point source will converge to a single focal point on the camera. Therefore, this angular ray information is lost, and imaging 3D space requires scanning axially and can be time-consuming. So in light field imaging system, by converging the ray bundle separately into different focal points, and this angular ray information is preserved, and imaging 3D volume can be captured in a single shot with high temporal resolution. So just to give you guys a dummy example, the conventional imaging system is like looking at the 3D sample from one projection view, whereas in the light field imaging system, you get this different angular projection views simultaneously on the screen in one shot. So without scanning, light field can achieve really fast 3D data acquisition speed, sometimes as fast as the camera frame rate up to like 80 to 100 volume per second. And then the 3D information can be recovered later after acquisition using standard 3D reconstruction algorithms such as Richardson deconvolution. So currently, light field has been applied in various imaging scenarios, ranging from cell imaging, organoids to zebrafish brain. However, these platforms are typically optimized for each specific scenarios, not really designed to accommodate different scenarios in one platform. So in the first part of my thesis, we aim to solve this issue and improve the versatility and ease of use of the light field imaging by combining it with a 2D wide field microscopy system. So our goal is to implement a all-in-one versatile light field microscopy platform that can accommodate bioimaging applications across different scales. So this is inspired from the conventional wide field microscope system, where you can achieve this so-called multi-purpose or multi-scale imaging with different objectives housed in a rotating turret. So this is a very common structure in most of the commercial microscopes. So what we did was simply attach a customized light field module to a commercial wide field microscope body. And in this way, we can take advantage of this automated turret to easily switch between different imaging scenarios by simply change to a different primary objective, which allow us to achieve this fast volumetric light field imaging across multiple scales. So the experimental setup is also very straightforward. You just need a camera, a microlens array, which is basically an array of mini lenses, and to capture multiple angular projection views, a tube lens for a relay, and a field stop to control the field of view. All of them are commercially available. So by using all commercially available components in the light field module, with the objectives we have in the lab from 60x to 10x, we were able to achieve imaging applications from cell biology to system biology. And moreover, the built-in flexibility and ease of use of the commercial microscope body makes the light field module accessible and user-friendly. And so today I'll showcase one very exciting result, which is to use 30x for imaging calcium dynamics in mice islet. So the islet is this globular structure of diameter around 50 to 80 micron, composed of the pancreatic cells, which is around 10 micron. So here we label the beta cell with calcium indicator GCAM6F. So this is a collaboration with a very talented graduate student called Janelle. So she will harvest the islet from the transgenic mice, isolated and rested in media at final glucose concentration for 5 days prior to imaging. So for this experiment, we recorded 70 seconds with frame rates at 20 volume per second. And as we can see here, with the high volumetric imaging rates, we were able to track these really fast calcium dynamics traveling across the whole islet in 3 dimensions. So to highlight this process, we computed the temporal difference movie of the 3D reconstructed volume. So basically, we just compute the difference between the consecutive frames to highlight these dynamical changes. So this allows us to see how these calcium signals propagate throughout the entire islet. So here the grayscale shows the average intensity over time, which serves as a structural background. And then the temporal changes are highlighted in yellow color. And as you can see here, within just a few seconds while we're talking, there are already a lot of these flashing events happening within the islet. So if we just ignore the 3D context for a moment and only look at the average intensity of the entire volume over time, which is similar to a EEG measurement, and we can observe this seemingly very repetitive spiking events. However, to take a closer look, we picked 3 events labeled E1, E2, and E3, and with each one for 1 second from the onset of the event. And then plot the color-coded depth image from minus 43 micron in cyan to 43 micron in red. And we can see that first, at t equals 0, in the first column, different calcium events initialize at different locations. Then, comparing vertically in each row, we can see that these spatial activations change at a millisecond scale. Then, comparing vertically in each column, we can see that these propagation patterns actually differ between different events. So they're not just repetitive oscillations, they're actually quite distinct from each other. And this is actually quite an exciting result because the diversity of these flashing dynamics is an area that remains underexplored and could potentially provide new insight into islet function. And we hope that as live field imaging enters into the islet research community, it will deepen our understanding of islet biology and diabetes and ultimately contribute to the development of new treatments. So apart from the islet, we also captured seizure propagation throughout the zebrafish whole brain using the 10x as the primary objective, and membrane protein dynamics in single cell using the 60x. So far, we've shown that a 2D wide field microscope enhances the versatility and usability of a 3D live field imaging system. And from there, we asked the reverse question: can the additional live field channel also enhance 2D wide field imaging? So this idea inspired the second part of my thesis, where we use the 3D imaging capability from the live field channel to improve the image contrast in 2D wide field imaging. So we know that a major challenge in 2D fluorescent microscopy is to reduce out-of-focus light above and below the focal plane. Specifically, in wide field microscope, the entire sample volume is illuminated during imaging. Light source at the native focal plane will be focused onto the camera, while the light above and below the focal plane will be out of focus. So these out of focus lights contribute to a blurry background to the focal plane image, which is something that we really want to get rid of. So we proposed a live field enhanced microscopy method. The central idea is to remove these out of focus background light above and below the focal plane using the 3D information captured from the live field channel. So in this way, there's no scanning. So since both live field and wide field system are inherently scanning free, so suitable for dynamic recordings. Also, the live field module is compact and can be easily constructed with good portability as a commercial add-on. And in particular, it provides sample-specific information in post-processing without imposing general assumptions on the sample, which is commonly done in standard wide field deconvolution methods. So to implement this concept, we'll take a wide field image and a live field image simultaneously, and then in reconstruction, we developed a joint deconvolution algorithm that combines the wide field image with the live field image for the 3D deconvolution. And in this way, the 3D information provided from the live field channel will enable the deconvolution to correctly reassign the background light back to its original plane away from the center focal plane. Therefore, we can just take out the center plane, and it is expected to give us a contrast-improved wide field image. So we validated this concept through simulation using a light sheet acquired 3D volume of silverfish brain vasculature as the ground truth biological data. So here the fish was at 5-day post-fertilization labeled with green fluorescent protein at the vasculature, and each slice was sampled at 1 micron Z-step. So on the left here is the central Z slice, which serves as the native focal plane image. And on the right is the full 75 micron depth volume, spanning plus or minus 37 micron above and below the central plane. So by forward convolving the ground truth volume with the system points by function, we simulated both the live field image and the wide field image of the 3D sample. So here we designed the system so that the depth coverage of the live field matches the sample thickness, meaning that we will be able to capture the full background contributions from the sample. And as you can see, this wide field image appears quite blurry due to out of focus background light from over 70 micron depth projecting all onto the focal plane. So for a quantitative comparison, we define the image contrast as the image standard deviation over the image mean, which represents luminescence difference over the average luminescence. So the rationale behind it is that a small difference is neglectable if the average luminescence is high, and the small difference matters if the average luminescence is low. So this definition is commonly used in image processing. So from the result, we can see that standard wide field deconvolution using information only from the wide field channel improves image contrast over the raw wide field image by roughly around 25%. Whereas in case of our live field enhanced microscopy method, jointly deconvolving the live field and the wide field images together improves the contrast by -2%, which is an additional 66% over the standard deconvolution method. So this shows us that incorporating 3D information from the live field channel is more effective at removing background signal from the focal plane image. So in summative, we developed a live field enabled microscopy framework that unifies both 2D and 3D imaging within a single platform. On one hand, we integrated live field imaging into a commercial live field microscope system, allowing for fast 3D bioimaging across various scales. On the other hand, we showed that 3D information from the live field imaging can also enhance image contrast in 2D wide field microscopy. And looking ahead, we envision that future wide field microscope system will have the option to integrate a live field imaging module, enabling not only fast dynamical imaging in 3 dimensions, but also enhanced 2D imaging contrast. So in the end, I would like to thank Matt for hardware support, Sebastian for providing the fish sample, then Janelle for the eyelid sample, then for the cell sample, and Scott and Ty for the mentorship. And last but not least, everybody in the facial.